Assalamualaikum warahmatullahi wabarakatuh, and a very good day to my respectful panel, TS Dr. Aisyah binti Muhammad Rehan and also TS Dr. Siti Aisyah binti Mansur. My name is Pushpa Syakilla binti Azni with metric number IDAN 190031. First, I would like to introduce my beloved supervisor, TS Dr. Nur Akmazila binti Muhammad Fauzi, and also my co-supervisor, Professor Madya Dr. Anzasari binti Muhammad Qasim, who have guided and supervised me along this journey. Today, I would like to present a proposal for my bachelor degree project entitled Anti-Inflammatory Study on the Soxhlet extracted Chromolaena odorata and Azadirachta indica leaf as a potential wound healing agent. In order to complete the proposal, a guidance outline has been created. This presentation will be included with introduction, literature review, methodology, expected result, and lastly conclusion. Without further ado, let's proceed to the introduction. The first part of introduction will be covered on research background. In this part, there will be a short brief about the plant chosen, the anti-inflammatory agent, and also the protein denaturation assay. Chromolaena odorata is a herb that usually known as pokok kapal terbang. Based on past research, this plant contains properties such as antioxidant, antimicrobial, and anti-cancer properties. The next plant chosen is Azadirachta indica. Is also a herb that usually used in traditional medicine. It is known as pokok semambu. This plant has been proved to contain properties such as antioxidant, antibacterial, and also antifungal. Anti-inflammatory agent is a substance that helps to reduce inflammation. Inflammation is a condition where the wound swells up and becomes red. And lastly, protein denaturation assay is an in vitro method to study the anti-inflammatory activity of plant. Protein denaturation has been well correlated with the occurrence of inflammatory response. Several problems are also arise from the study. Firstly, even though we have known both Chromolaena odorata and Azadirachta indica are widely used in traditional medicine because of their richness in bioactive compounds, the research about these two plants are still lacking. Moreover, in wound healing, the study of anti-inflammatory activity for both plants are also still limited. Lastly, the use of harmful chemical substances as a wound healing agent. These harmful chemical substances could be harm to people's health. Thus, it is important to find out the potential natural ingredient that can be used as a wound healing agent. Next, this study is aimed to study the anti-inflammatory activity of Chromolaena odorata and Azadirachta indica leaf extract. Next, to determine the optimum condition of Soxhlet extraction of Chromolaena odorata and Azadirachta indica leaves. And lastly, to apply the extracted Chromolaena odorata and Azadirachta indica leaves extract in formulated cosmeceutical products and determine their anti-inflammatory activity. In order to achieve the objective above, this research will be extended to the more specific study scope. Firstly, the plant samples will be collected from Nasua herbs and spice farm in a fresh condition. Next, the process condition of Soxhlet extraction will be tested at different parameters. After that, the study of anti-inflammatory activity of both samples will be tested by using protein denaturation assay method. After that, the effectiveness of anti-inflammatory activity for both plants as a wound healing agent will be compared. After that, the application of both extract as wound healing agent will be tested for the production of cosmeceutical products such as lotion bar and baby oil. And lastly, the result obtained from anti-inflammatory analysis will be further analyzed using ANOVA and Microsoft Excel software. Okay, now let's move to the literature review. The first topic in literature review is about the two plants chosen. Firstly, Chromolaena odorata. Chromolaena odorata is originated from Central and South of America. This plant belong to Azadirachta family and can grow in various types of soil except for waterlogged and sandy soils. These plants contain components such as tannins, alkaloids, and also flavonoids. Different parts of leaves shows different application use. For example, the root of this plant can use as analgesic and involved in the treatment of the skin. Meanwhile, the leaves can help to cure sore throat and flu. The flavonoids is one of phytoconstituents that exhibit antiviral properties that help to lower the intensity of common flu and short duration of a flu symptom. The next plant, which is Azadirachta indica, is originated from Indian subcontinent but mostly growing in Asia and Africa certain areas. It is from Meliaca family and it contains complicated components such as nimbidine, nimbolite and also limonite, which is play a major role as anti-inflammatory components. Just like Chromolaena odorata, different parts of Azadirachta indica also shows a different use. For example, the extract of bronchopeptic ulcer, the leaf help to treat skin disease, and lastly, the seed will act as a pesticidal. In today's market, there are a lot of anti-inflammatory agents that use chemical substances that could be harmful to people. Natural anti-inflammatory can be extracted from plant materials such as herbs and spices. Both Chromolaena odorata and Azadirachta indica contains components such as quercetin. Quercetin has been described as a long-acting anti-inflammatory with strong anti-inflammatory properties. The next topic that will be reviewed in the literature review is Soxhlet extraction. Generally, extraction is a process to separate desired product. In this study, the leaves of Chromolaena odorata and Azadirachta indica will be extracted. Soxhlet extraction is a solid liquid separation process where object in solid phase will be placed in contact with solvent. In this study, different parameters will be used to compare the extraction yield, where solvent used are 95% ethanol and distilled water, while treatment time will be used is 1 hour and 6 hours. So, from different extraction yield, we can also observe the effect of it on the anti-inflammatory activity. The figure shown in the slide displays the configuration of Soxhlet extractor. It consists of condenser, extraction chamber, thimble, sniffer arm, extraction solvent, and lastly boiling flask. The sample which we already turned into powder will be placed in the thimble. Meanwhile, the solvent will be placed on the boiling flask. The next topic that will be explained is protein denaturation. Protein denaturation is a biochemical process that involves the disruption of covalent bond in protein structure. Changes in protein structure are frequently accompanied by changes in attributes such as chemicals, physicals, and functional properties. For example, pH and heat. In this research, protein denaturation is used to determine anti-inflammatory activities. It is because the potential for a sample to inhibit protein denaturation may prevent the inflammatory illness. Wound healing is the body's natural response to tissue damage. It consists of meticulous and highly programmed phases, which are homeostasis, inflammation, proliferative, and lastly, remodeling. Various herbs composition helps to expedite wound healing. Properties such as antioxidant, antimicrobial, antifungal, anti-inflammatory, and also antiviral are related to the wound healing process. For example, antioxidant had a role to aid in wound oxidative stress control and hence speed wound healing. In methodology part, I will explain shortly about how will the method used is carried out in order to study the anti-inflammatory activity of Chromolaena odorata and Azadirachta indica leaf extract. Firstly, the sample will be cleaned and dried for 2 hours after collecting it from Nasuha form. Next, the dried leaf will be grinded to turn it into powder form. Extraction will be carried out using Soxhlet extraction in different parameters as mentioned in the previous slides. The extracted leaf tanks will be undergoes protein denaturation assay to study the anti-inflammatory activities of the leaf extract. Each test tube will be containing BSA solution, phosphate buffer saline, and sample in different concentration. BSA will serve as protein in the experiment. Next, to determine the percentage inhibition of protein denaturation of the leaf extract, the turbidity of the sample will be measured by using UVB spectrometer at 660 nanometer. Finally, the measured data then will be further analyzed by using Microsoft Word, Excel, or ANOVA. From the research work, it is expected for both Chromolaena odorata and Azadirachta indica leaf extract to have good potential as natural anti-inflammatory agent in a wound healing. Moreover, considering the traditional use of these plants, it shows that both plant is very effective in treating wound. Furthermore, the different parameters used in Soxhlet extraction also will give different extraction yield and also affect the anti-inflammatory activity. Thus, it is believed that the leaf extract of both plants can be used as a natural anti-inflammatory agent to replace the use of harmful chemical agent that will give side effect to consumer. To conclude this presentation, this research is focused on the studying the anti-inflammatory activity extracted from Chromolaena odorata and Azadirachta indica. Besides, the inhibition of the natural protein indicates the anti-inflammatory activity. Moreover, different parameters used in Soxhlet extraction method will affect both extraction yield and anti-inflammatory activity. And finally, the effectiveness of both plants to act as natural anti-inflammatory agent in wound healing can be estimated. That's all from me. Thank you.